it changes the onus away from you being predisposed to getting UTIs, for example, because it's actually this exact same infection which is not being cleared correctly.
Hi, welcome back to the Live UTI Free channel. My name is Melissa, and today I'm talking to Nick Parkinson, a PhD in molecular genetics and scientific director at Systems Biology Laboratory, a UK-based non-profit research lab. In part one of this interview, we'll discuss what healthy urinary and vaginal microbiomes look like, how this can change with infection, and whether probiotics truly make it to their intended destination. If you enjoy these videos, think they're important, and want to support what we do, make sure to hit subscribe and tick the bell so you'll be notified of any new content we post. Thanks again for joining us on this journey to making change in women's health. Today I'm speaking with Nick Parkinson, who has a PhD in molecular genetics and is the scientific director at Systems Biology Laboratory, a UK-based non-profit research lab. Thanks for joining me to answer our community's questions.
It's lovely to be here. Thank you very much for the invite.
Yeah, you're welcome. Can you share a little bit about your background and why you became interested in UTI research?
Absolutely. So the lab that I work for, and I've been here for about 12, 13, 14 years now, time goes quite quickly, is a very small not-for-profit research group, which is run by our philanthropist, Mike Fisher, who funds us exclusively. And we have a remit, really, to try to leverage our specialism in looking at DNA sequences and different types of sequencing technologies to try to apply them to help with healthcare issues which are currently under the radar or invisible. We spent a long time looking at cancer immunotherapy and various other projects before we came across the UTI issue and decided that we were going to try and come up with our own solution. And the reason we got into that really is a personal one. And my daughter, from the age of about 3 to the age of about 7, suffered from recurrent UTI, but obviously being very young, she didn't know how to vocalize that or how to express that. But as parents, and obviously very concerned parents, we recognized a lot of the symptoms with bedwetting, behavioral change, fever, all those kind of things. Very consistent with it being a UTI. The GP over that 4-year period agreed with us, but unfortunately, which was quite a new situation for us, we were confronted by this barrage of escalating consultants telling us that there was nothing wrong because the dipsticks and the culture were coming back negative, and that it was probably a developmental thing that she would grow out of. And so one day I came in very frustrated to the lab, and I sat down with the director and I just said, look, can we do something about this? I've got some ideas. Let's go to the lab. Let's see if we can do some DNA sequencing. And so very quickly we got the team together and we came out with a rudimentary protocol procedure, which allowed us to take one of her urine samples and to harvest the DNA from it, sequence it, and then work out from the sequences of DNA that we got back what might have been in there. And we were really surprised to find that within a week, we had a really strong signal for a bacteria that none of us had ever heard of before, which is called a Tinatignum charlii. And we went back to the GP, and the GP was very understanding. And she managed to lean on their local pathology department to try to reproduce the findings. Now, we got very lucky because the pathology department saw it as a bit of a challenge. And so they tried to grow this bacteria and changed all of their normal standard operating procedures to meet the exact culture requirements of this bacteria, and managed to do it and showed that there was tons of it there. We then dipped into the literature and found very quickly that it was a very simple amoxicillin course should get rid of it, and it did. And I'm very happy to say that, you know, she's now 14 and she's never had another episode since. And we kind of sat back and we just thought that was really easy. You know, even if 1% of all UTI, chronic UTI or recurrent UTIs are that easy, that's still a hell of a lot of people. And it opened the doors for us on an entire community of sufferers globally. And what is a horrendous invisible disease, which we had no real idea about until we had personal experience. And we made the decision right there and then that actually we're going to drop all of our other research and we were going to start to try to refine this process and to see what we could actually do with this to actually provide a new tool And that's really what we're focused on doing, is providing a tool which is fit for purpose, to allow medics to actually make informed decision rather than using these Victorian or even medieval techniques that they're currently using, which just don't work. And I think universally that's becoming more and more accepted.
Yeah, it's quite clear to the patient community anyway, waiting for everything else to catch up. But it'd be great to get an overview of the type of testing that you are providing now in the UTI space.
So as I say, we started out by just coming out with a rudimentary test to see what DNA was in there. There are other tests available now. There weren't at the time, but we really wanted to move away from culture. We felt that culture had the risk of biasing what was in there and what was detected, because really it needs to grow to be able to see it. And it's catch-22 situation, really. You don't know what you're trying to grow until it's grown, and you have no idea what you might be missing.
Yeah.
And bacteria are very fickle. Some of them will grow easily. Some of them won't grow well, you have to provide them with incredibly specialist conditions before they'll even think about growing. And so culture seemed to be a dead end for us. Equally, PCR, we've done a lot of work with PCR in the past, and we recognize its limitations, the fact that it can be overly sensitive sometimes and actually find signals when signals aren't really present. And it can also distort signals to a degree, because it's effectively like photocopying the same page again and again and again and again and again. And so we decided to try to come up with a technique which was completely free of culture and completely free of PCR, and to take out all of the middlemen, really, effectively, and just go straight to the sample, extract all of the DNA we could find in there, and sequence it. And we spent a long time realizing that it's not how you produce the data. The skill really is in how you interpret the data and how you match the data to the databases, and how you construct your own databases. And what I mean by a database is that all bacteria have their own unique code, DNA code, and you can use it like a fingerprint to identify them. If you find a fragment of DNA in your sample and you read it and you go, okay, right, well, what does this match to? And as long as you've got a really good, clean database of robust genetic comparisons, then you can find out quite quickly and with some degree of certainty where that came from. And that way you can say that that bacteria was present in that sample. So that's what we do. We spent a long time working out how best to extract the DNA to make sure that we collect all of the DNA that's in the sample. And incidentally, that doesn't just stop at bacteria. It's mainly targeted at bacteria, but we also deliberately disrupt and remove DNA for yeast to collect that. And along with that, we also managed to collect DNA for viruses, and also other elements of bacteria, which most people are completely unaware of, such as plasmids and phages, which I might talk about a little bit later on if people are still with us. But we collect all of that DNA, and then we sequence it using quite a novel version of sequencing technology, which has been technology for sequencing DNA has been around for 20, 30 years now. But this is a newcomer on the block, and it's called nanopore sequencing, and it allows you to end up with bigger chunks of DNA in one go. And it's perfect for this kind of application, because it means where you're trying to match an individual strand of DNA to a reference, the longer that strand is, the better the chances that you're going to find a unique match to a reference. So that's what we do. We take all of the DNA out, we sequence it, and then we spend a time with our bespoke bioinformatics software to actually come out with really clean comparisons, which have a lot of noise cancelling in them. I think I've been thinking about this for a while now. I think analogies are really useful in this. It's quite complicated stuff. It's technology and ways of talking that everyone in the field are very comfortable with. But I think for most customers and most people who are suffering from UTIs, it probably sounds like double Dutch. And I'm a puzzle solver. I've always been a puzzle solver, and I love a jigsaw, especially at Christmas. And so my analogy is going to be based on jigsaws. So I have been to a couple of amazing jigsaw shops. There's one in London that I go to, which has thousands and thousands of different jigsaws in it. So Imagine you are standing in a shop which has 4,000 different jigsaws
Mm-hmm.
in the shop, and you are handed a single piece of a jigsaw, and you are asked to say which jigsaw that's come from the 4,000 that are on the shelves. Now, that's quite a task. And that's the same task that we're given constantly for trying to match our DNA back to our references being the different types of jigsaw that are present. Now, we're not the only people that use DNA to try and match and to try and identify what might be in a sample. There are other techniques available. And the most popular one is PCR. There are lots of commercial companies out there that use PCR. And now PCR will only really tell you whether a bacteria is present or absent. And it's limited to looking for a predefined list of bacteria, which is normally, most commercial services are about 10 or 15 different bacteria that are looking for, you know, the usual suspects, E. coli, Faecalis, all that kind of thing. But they'll only tell you if they're in there. They won't tell you what else is in there. And so PCR is quite limited. The best alternative is something called 16S RNA. And that is a next generation sequencing process like ours, but again, is quite limited. And it's a blend of PCR and actually looking at the sequences. Now, 16S RNA doesn't really take full advantage of all of the DNA that's present. It only looks for a particular subpart of the DNA. And it works out to be about 1/10,000th of the total DNA that a bacteria has. So to stretch our analogy a bit more, let's imagine that all of our jigsaws are 10,000 piece jigsaws, and you're handed one piece, and you've got to match it. Now, that's fine if your piece has something really remarkable on it, like a face or, I don't know, something that really stands out. But if it's a piece of sky or a piece of cloud or whatever, if there's no real obvious markings on it, you may find that you can only really whittle it down to maybe 100 or 1,000 different jigsaw puzzles that are in there it could have come from. Now, that's the problem with 16S RNA. It's not always possible to make a specific match to an individual bacteria. You may be able to say that there are lactobacillus present, but not necessarily give the actual species. There are other limitations as well. 16S RNA only works on bacteria, so it doesn't matter how much yeast you've got in there, it'll never find it. So how does that compare to what we've done? Well, because we use nanopore sequencing, which is very different, it allows us to use all of the DNA that we capture, which is all of the DNA in the sample, not just this little 1/10,000th bit. But it also allows us to cheat, which is a fantastic thing to do. It allows us to produce jigsaw fragments which are maybe 10 times, 100 times, sometimes even 1,000 times bigger. The actual jigsaw puzzle itself is still the same size, but we now capture more of the picture in any one puzzle piece. And so now suddenly you may have a 1/10 of the entire puzzle piece in your hand. And so it makes it very easy to find which of the 4,000 puzzles it came from. And you can match it very, very quickly and very specifically. So that's what we do. But we don't just go into these puzzle shops with just one piece like 16S RNA. It's not just a single piece. You go back into your DNA that you've harvested from your sample and you repeat and repeat and repeat, and you keep sequencing different fragments to build up a full picture of what was in there. And typically we use about 500,000 individual fragments of DNA for every sample that comes into the lab. And so you can imagine that standing in that shop with 500,000 different fragments becomes quite boring after a while. But the computers and the code that we've developed turn it over incredibly quickly. 16S RNA really struggles to do it quickly because the matches are much more laborious to produce. Whereas our code and the ability to cheat means that we can turn it around very quickly, which is also a big advantage for us. It means that once we've extracted the DNA and we've processed it to a point where we can put it onto the sequencing machine, we can normally get an idea of what's in there within only a few hours. We typically leave the sequencing overnight to complete, and then all of our computation happens the next day. But from the sample coming into the lab to when we post results is typically 14 hours or less. We like to say 24 hours or 48 hours because we don't want to leave we would have to leave a bit of wiggle room in case something goes wrong and we need to repeat it. But typically it's incredibly quick compared to 16S RNA sequencing, which will take a week or maybe 2 weeks to turn around. Now, that's quite important if you're sitting at home and you're in excruciating pain and you just want to know what it is that's causing your UTI. And so we think we have a huge benefit over other sequencing technologies in that respect alone. But we also have this other really important advantage in that because our pieces are really big, it means that, and because we use all of the DNA, it means that quite often we're able to complete the entire jigsaw for individual boxes and pictures, which 16S RNA can't do. It's only looking at this 1/10,000th, and that's all it can ever provide for any one particular puzzle. But it means that we can fully recreate all of the DNA that makes up a particular bacteria in a particular person. And bacteria, like humans and anyone else, will have slight changes in their DNA from type to type. And this can be within the same family. So, for example, an E. coli that I might get as an infection might look very different to an E. coli that you might get for an infection. They still are similar enough to both be E. colis, but if you really dig down deep enough into the 3 million base pairs of DNA that make up its DNA, you will find individual differences, which means that actually the strain of DNA, the strain of E. coli that you have is probably unique to you. And this is something that's not no other technique is really able to see that, but we are, and it's really important because different strains of E. coli, for example, are known to act differently, behave differently. They have different characteristics in terms of whether they are likely to be pathogenic or not. And so we can quite quickly take an E. coli which is found in your sample, and we can recreate the whole genome, and then we can compare that genome to the other ones that we know, the other strains that we know about, and with known characteristics. And we can make very firm predictions over what the E. coli that you have is likely to do in terms of behavior and which one it most resembles. That's really important information when you're trying to treat this and when you're trying to make a judgment call as to whether this might be causative to your symptoms, which is obviously a big issue. So we really feel like we got a lot of advantages over any other method that's currently out there, not just in terms of speed, but the depth of information which we actually can create. It really is individual healthcare. And actually, to an extent, we're running to keep up with the technology, and we know that we can exploit the technology a lot more than we already are. So, for example, again, we have within bacteria, there are also these accessory bits of DNA that they can share quite quickly between communities of bacteria, which can change their characteristics. We capture those as well. And we can quite quickly work out how they might change the character of that bacteria. And even bacteria can get infections themselves. They can get infected by viruses, which we also capture. So we can also tell whether the virus that's infecting you has in its own respect got its own infection. Now, these systems seem quite academic, but they're really coming to their own when you're trying to work out whether your infection is a new infection to you or whether it's simply a reemergence of an infection that you've had previously, which has just been incompletely treated. And that can change, I think, the landscape of diagnostics a little bit, because it changes the onus away from you being predisposed to getting UTIs, for example, to actually it's the medical interventions that need to be rethought, because it's actually this exact same infection which is not being cleared correctly.
Speaking of persistent infection, can this type of testing detect whether a biofilm is present?
It's a really good question. And obviously, there's a lot of onus at the moment on biofilms and the mechanisms of pathology, which aren't fully understood by any means. And I'm certainly not the person to be discussing them. But we've done a lot of work to try and work out exactly what it is that we're detecting and where the bacteria are coming from that are in our sample. So we know that there are going to be some bacteria that are free floating. Let's start with urine samples, for example. There will be a percentage of bacteria that are free floating, but for the vast majority of bacteria that are in a UTI or in a bladder, they generally need to be anchored down to the surface of the epithelium, the skin that lines the inside of the bladder. Otherwise, they'll get washed out quite quickly. And a lot of these bacteria are able to do that, and some of them are even able to bury themselves into the skin cells that are lining the epithelia itself. We know from the work that we've done in the lab that a lot of the bacteria that we're seeing are not just the free living ones, but we find the bacteria that are attached to skin cells which are sloughed. And skin cells are actively sloughed by the epithelia of an infected individual as a response really to that infection. You get local inflammation, which promotes sloughing and loss of skin cells. And with those skin cells, you lose a lot of the bacteria that are attached to them. Now, the way that we process our sample means that we release those bacteria from both the surface of the skin cells and also if they were buried inside the skin cells. And we suspect heavily that the same is true with biofilms, which are likely to be attached to the outside of those skin cells as well. So we haven't got any direct evidence for it, but we suspect strongly that regardless of whether the bacteria is free living or attached or buried within inside a skin cell or even within a biofilm attached to the outside of a skin cell, we think we're seeing all of them. Certainly a large percentage of them, and certainly a lot more than you would see on a culture or a dipstick.
Sure. And you mentioned that the biofilm is kind of the normal state for bacteria in the bladder. Have you done research into healthy controls and what the urinary microbiome might look like?
We have, not in the context of how and why, what structures the bacteria might be living in terms of biofilms, but we absolutely have. As a group, we're all quite long in the tooth scientists now. We've had quite some careers in science, and we're all academics at heart. And so we're very proud of this new technique, but ultimately we love the idea that we can continue to do research in the background and use this as a tool to help promote understanding of what's going on. And part of that is being able to compare the results that we're getting from people with symptoms to people who haven't got symptoms. And we need to understand what a normal, healthy urobiome looks like. As Alan Wolf, many years ago showed, there is no such thing as a sterile bladder. And so we need to understand what bacteria are present, what their levels are, how dynamic that is, and how, or is it relatively stable? And what happens over longer periods of time when other species try to muscle their way in, and how they might do that. And ultimately, how any of this then relates to symptoms. But yes, we have done quite a lot of work on healthy controls. It's something that we're trying to expand all the time, which enables us to almost have the counterpoint to compare our results that we get from symptomatic individuals.
Which brings me to the next question. Are there organisms that you see frequently in recurrent UTI or chronic UTI patient samples?
There are. We see, first of all, it's an interesting field at the moment because there's a lot of flux, there's a lot of change, there are a lot more different methods coming in, certainly academically, to peer inside what's happening in a bladder using DNA technology similar to ours. But most of them are academic tools, and so people are constantly reevaluating what's actually going on inside the bladder. And there's we generally see a lot of what we would consider to be uropathogens. So these are species that have a known history link with causing pathology in urine. But even that term itself is quite vague, and it's very hard to actually pin down what might be considered a uropathogen, what might not be. So we can start at one end of the spectrum. Something like E. coli or Enterococcus faecalis is something that most people would probably agree that if you've got it in large quantities in your bladder and you've got symptoms, it's probably linked. We do see a lot of people who have E. coli. We do see a lot of people who have Enterococcus faecalis. And the usual suspects you would hope would be picked up by regular screening in clinics up and down the country. It's a real worry that a system, so the culturing-based and dipstick-based systems that are used in the NHS, which are largely focused on detecting Enterococcus and E. coli, even those seem to fail to find E. coli and Enterococcus in some samples, which is even more concerning than I think as a picture than we ever anticipated.
Yeah.
We are also, because we're so new, we're also very mindful of the fact that we're seeing a lot of patients who are really, they're very desperate. They've tried every test known, to humanity. Often they've been trying to get a resolution for their UTI for years, maybe decades, and they're suffering tremendously. And so we think that currently the first people that are finding us and using our technology are probably quite a niche group of people who are really long-term recurrent sufferers. And so it's quite hard to know how much of what we're seeing is a reflection of the larger population of UTI sufferers in the UK, or how much of this is really just a reflection of these people who have been failed for so long. But yes, we see a lot of the usual suspects, but then we also see a fair number of unusual suspects as well. So we see quite a lot of bifidobacteria of different types, bifido breve, bifido longum, we see quite a lot, and in high levels as well. But as they're not known and accepted uropathogens, it's very hard to be able to point to them and say these are definitely what's causing your symptoms. And again, the whole thing loops round on the question of what is causal and what is not causal, which is a question which has been on the cards and in the UTI communities increasingly over the last few years. And it's an absolutely valid question to ask. And I think something that's muddied the waters slightly is that many of the early DNA-based technologies like 16S RNA, they do seem to be prone to producing false positives.
Okay.
And that means that it's now very much accepted that a lot of uropathogens are, it's possible to have them at low levels in healthy controls. So E. coli, for example, it's now accepted that just having E. coli in the bladder doesn't necessarily mean that you're symptomatic or that it's that you're likely to have an episode. We don't see that as much, to be honest, and we are starting to question how much of that has come from 16S RNA's slight ability to produce false positives over. There are other benefits to our system that we've spent a long time to make sure that we calibrate our systems to allow us to actually give a very accurate level of each of the bacteria that are present. And so when we do see E. coli, it's at incredibly low levels. And it's at those low levels, it's very hard to know whether they really do come from the bladder or whether they've just been picked up as this stream of urine has been collected either on the skin locally or the way that the sample has been handled.
Yeah.
So they're not reliable indicators at the moment. But yeah.
Okay. And do you also find fungi in urine samples frequently?
We do.
Just the typical Candida species or other things beyond that?
We see a lot of samples that have Candida albicans, which is the most common. But we also see quite a number of samples which have Candida glabrata, which also, the problem with a lot of these species that they get renamed every now and then. And so it also has a particularly long and convoluted name, which is the more modern name, but I'm not going to attempt to pronounce it. I'm going to call it Candida glabrata at the moment. It's effectively the evil twin of Candida albicans, because unlike albicans, where there are lots of off-the-shelf thrush remedies that you can use and treatments that you can use, glabrata seems to be largely resistant to most of them.
Right.
Which is why it's important to know which one you have. And it's where most testing systems really fall down. In our testing system, even though we're harvesting all of the DNA that's in the back in a particular sample, we're only as good as the references that we're comparing them to. And so we're very aware of the fact that there's a small chance that there might be a bacteria that's in there that we don't have a reference to. So if we don't have the jigsaw box to match it to, we can't ever make that comparison. And so we've worked very hard focusing mainly on our bacterial libraries to make sure that we have as wide a range of bacteria present as possible. But the yeast has lagged, to be honest. And it's something we've really been working on hard recently. We're now up to 48 different yeast species, which covers virtually all of the yeast species that have been reported to live on or in a human.
Right.
But we're on the cusp at the moment of expanding that out to a full 300 different species.
Okay.
And so we may find other, in very rare instances, we may find other yeast present. But at the moment, it's generally glabrata and albicans are the two main suspects that we find.
Okay. And I know you offer both vaginal swab testing and urine testing. Do you find that you see similar organisms across both of those types of tests in the same person?
Absolutely. And we first started out looking or designing our protocol to look just at urine. And then we very quickly realized that actually by using a simple vaginal swabbing method that we could actually find out what was going on in the vaginal microbiome at the same time. And we were really amazed to see quite how similar they were. The levels that we get back from the vaginal swab samples are hugely different, massive amounts of cells that we get back. But then that's largely to do with the fact we're using a swab as opposed to looking at a relatively dilute urine sample. But in terms of the actual bacteria that we see and the relative positioning of those bacteria in terms of which are the most prevalent down to which are the least prevalent seems to be really very similar in both situations, which is why now we really try to push wherever possible that people who have symptoms but don't really understand what might be causing those symptoms really should try doing both tests at the same time, which is why we offer both of the tests as a slight discount if you buy them both at the same time, because it really does make diagnosing or at least identifying a bacteria that might be causing your symptoms so much easier. If, for example, you've got massive amounts of E. coli or Enterococcus in the bladder, but you don't have any vaginally, it really helps with clinicians using our tool, where they're actually trying to come out with, on the balance of probabilities, what might be causing it. And that's really all clinicians are ever able to do. Diagnosis is always going to be a balance of probabilities judgment call based on the information they have. All we're trying to do is give them all of the information that they need. But equally, we find something quite startling, where we've seen plenty of patients who have got large levels of Enterococcus faecium or faecalis, for example, in the bladder. And that's all you would see if you tested the bladder alone. And you would quite happily sit there and say, this is probably causing your UTI, you should get treatment for it. But when you co-profile the vagina, you also find that there's a massive residue of the same species there as well. Now, why that's important is that most treatments for UTI are using antibiotics which accumulate specifically in the bladder and are excreted through the kidneys. And so you're targeting the population of those bacteria that are in the bladder, but not necessarily doing anything to address the population that are in the vagina. Now, given that these populations are shared so readily and constantly, it means that when you finish treatment, you may well have cleared your UTI causing microbe in your bladder, but if there's a massive pool of it sat next door, it's very quickly going to come back. And so I think knowing what's happening across both systems is really important, but I think there's also needs to be a little bit of education on the clinical side as to how to treat these and how it really is. There's no point in stamping out one fire if you've got a burning building next door. You've got to be able to treat both at the same time.
Thanks so much for watching. I hope you found part 1 helpful as we discussed the urinary and vaginal microbiomes and the impact of probiotics. If you want to learn more about this or related topics, be sure to check out our other videos or head over to liveUTIfree.com for articles related to this video. We'll be sure to put some links in the video description. If you like what we're doing on this channel, be sure to hit subscribe here on YouTube and tick the bell so you'll be notified of our future content. Thanks so much for watching, and until next time, keep asking questions and pushing for better solutions.